Our immune system is a remarkable defense force. It protects us from a constant barrage of microscopic invaders. Among the immune system's many weapons, the complement system stands out. This system acts as a rapid response team, neutralizing threats and rallying other immune cells to action. The Mannan Binding lectin, or MBL, pathway is a critical part of this intricate defense network. Searching for suspicious patterns on the surfaces of microbes. These patterns are like molecular signatures found on the outer coat of many bacteria and viruses. When MBL encounters these patterns, it rapidly binds to them, triggering a cascade of events leading to the destruction of the invader. The MBL pathway is a crucial first line of defense against infection. Its swift and efficient action makes it a key player in keeping us healthy. In the following sections, we will explore the intricacies of this remarkable pathway. The complement system is a powerful weapon. It is a key component of our innate immune response. There are three main pathways: classical, alternative, and MBL. Each pathway recognizes pathogens uniquely but aims to neutralize threats. The MBL pathway is similar to the classical pathway. Both recognize specific molecular patterns on pathogens. The classical pathway uses antibodies. The MBL pathway uses Mannan Binding lectin, MBL. This difference in initiation is crucial. Both pathways converge at a critical point, leading to the formation of the membrane attack complex, MAC. This underscores the complement system's efficiency. Imagine the surface of a bacterium as a bustling cityscape. It has a diverse landscape of molecules with unique shapes and properties. Among these molecules, certain patterns act as red flags to our immune system. These patterns are known as pathogen-associated molecular patterns, or PAMPs. Mannan Binding lectin, or MBL, is a key player in this molecular surveillance system. MBL is a protein with a remarkable units that assemble into a bouquet-like structure. Each subunit has a carbohydrate recognition domain, or CRD, acting like a lock that can only be opened by a specific key. When MBL encounters a pathogen displaying these specific sugars, its CRDs bind to the sugars with high affinity. This binding triggers a cascade of events that ultimately lead to the pathogen's destruction. The binding of MBL to the pathogen's surface is not random. It is a highly specific interaction. This specificity ensures that the MBL pathway is activated only in the presence of genuine threats, minimizing the risk of attacking our own cells. By recognizing and binding to these pathogens, MBL effectively tags them for destruction by other components of the immune system. Section 4. Structural echoes. MBL and C1q similar forms for similar functions. Nature often reveals elegant solutions to complex problems. This is certainly true for the complement system. Two key players in this system, Mannan Binding lectin, or MBL, and C1q, share a striking structural resemblance. This resemblance reflects their common role in triggering the complement cascade. Imagine both MBL a common goal: to activate the complement cascade. This cascade is a series of enzymatic reactions that ultimately lead to the formation of the membrane attack complex, or MAC. The MAC creates pores in the membranes of target cells, leading to their destruction. The structural similarity between MBL and C1q highlights an important principle in biology: form follows function. This resemblance suggests a possible evolutionary link between the MBL and classical pathways, hinting at a common ancestor. Both pathways play crucial roles in immunity, activated by different mechanisms but sharing structural features. Section 5. The protease powerhouse, MSP1 and MSP2. Orchestrating the complement cascade. The complement system amplifies a small signal into a powerful immune response. This is achieved through a cascade of enzymatic reactions. In the MBL pathway, MASP1 and MASP2 are key proteases. These precursors circulate in the blood, awaiting activation. MBL binding to a pathogen activates MASP1 and MASP2. They initiate a cascade crucial for amplifying the immune response. This activation is tightly regulated to prevent damage to our own cells. Section 6. A cascade of destruction. From MBL binding to microbial elimination. The binding of Mannan Binding lectin, or MBL, to a pathogen is just the first step in a complex chain reaction. This chain reaction ultimately leads to the elimination of the invader. Once MBL binds to its target, the associated MASP1 and MASP2 proteases spring into action. These proteases set off a cascade of enzymatic pathogen. C3b attaches to the surface of the pathogen, marking it for destruction. This process of marking a pathogen for destruction is called opsonization. C3b also plays a crucial role in the formation of the membrane attack complex, or MAC. The MAC assembles on the surface of the pathogen, forming a pore-like structure that disrupts the integrity of the membrane. This disruption leads to the influx of water and ions, causing the pathogen to swell and eventually burst. Section 7. Conclusion. The MBL pathway, a vital defender against microbial invaders. The Mannan Binding lectin, or MBL, pathway stands as a testament to the elegance and efficiency of our immune system. It detects specific molecular patterns on pathogens and triggers their destruction. Acting swiftly, it neutralizes bacteria, viruses, fungi, and parasites. Deficiencies in MBL can lead to increased susceptibility to infections. Understanding the MBL pathway opens avenues for therapeutic interventions, ensuring our survival in a world teeming with unseen threats.